You may well ask, science at a literary festival, why? And I'm here to provide some answers and hopefully some entertainment. There are at least a few reasons to be here. One is to explain the relationship between science and politics. We're hearing a lot of politics today. I want to tell you something about the beauty of science, what science has uncovered about the way in which life forms work. And I want to tell you something about the utility of science. It's important that science produce results for the economy, for health, for the environment. And we believe as scientists that while we enjoy the excitement of making discoveries, we also believe that science is useful and that the utility of science is part of the reason why science does get support from politicians and from governments that allows us to receive money that supports the expensive work that we do. Now, what I'd like to do today, understanding the hour is late and we want to have some discussion as well, so I'd like to spend a little bit of time telling you about how the political support for science in the U.S., the country I know best, has made scientific life possible for me and for many others in the U.S. I'd like to tell you a little bit about what science has taught us over the last 50 years or so about how life works. I'm going to show you some images, even some movies, some animations that are quite brilliant. And then I'd like to tell you a little bit about how science has become useful by allowing us to understand a terrible set of diseases, cancers, in ways that is allowing us to improve the way we treat cancers and think about controlling cancer worldwide. I do have an ulterior motive. Bangladesh is a very interesting place. It's my first time here. I've spent a large amount of time in India, but I've never been here before. And I am deeply aware of the fact that unlike what most people tend to think of in the United States, that Bangladesh is a very populated country, 6th largest population in the world, and yet its contribution to basic science still seems relatively low compared to the number of people and the number of very smart individuals here who are doing great work, as we're hearing, in literature and arts. And I'd like to think that inspired by hearing some scientists talk at a place like the Dhaka Literary Festival, that there will be a greater interest in supporting basic science in this country. So let me begin by telling you something about my own perception of what science does for the world. When I was a college student at Amherst College in Massachusetts, after being in school for a week, the Soviet Union suddenly surprised the U.S. by putting a satellite into orbit around the Earth. This satellite was called Sputnik. It required a banner headline in the New York Times. It frightened the U.S. to think that a Soviet vessel was circling the Earth every 24 hours or so, and that the U.S. was now being challenged as a place where science and technology was dominant. And indeed, our president at that time, Dwight Eisenhower, and presidents to come, used this challenge to make deeper investments in scientific research and technology development, in teaching math and physics and chemistry and other sciences in high schools and in colleges. One of the things that was almost a direct result of Sputnik was John Kennedy's decision to say, we're going to send a man to the moon. That was an additional challenge to American science and technology, and indeed that ambition was achieved. Less well known to me, because it occurred when I was a very young boy, in fact less than a year old, was a deeper, earlier investment in the idea of science that came about at the start of World War II. At the end of October in 1940, when Europe was already involved in World War II, Franklin Roosevelt, then at the end of his campaign for a 4th term as President of the United States, came to the new campus for the National Institutes of Health, a place where I was trained. I've been the director of the institutes subsequently, but he came to the building where I later served as director in 1940 and made a very famous speech about the importance of investing in medical science. I'm going to play you a few things from
Dr. Thompson, life conservation.
Sorry. Let me start at a good place here.
Ladies and gentlemen, nowhere in the world except in the Americas is it possible for any nation to devote a great sector of its effort to life conservation rather than life destruction. All of us are grateful that we in the United States can still turn our thoughts and our attention to those institutions of our country that symbolize peace, institutions whose purpose it is to save life and not to destroy it. It is for the dedication of these noble buildings to the service of man that we are assembled here today. The National Institute of Health speaks the universal language of humanitarianism.
So, during World War II, science contributed a lot to the war effort. The improvement in radar, the production of penicillin, the production of the atomic bomb, and many of these things inspired Roosevelt to tell the director of the national science effort, Vannevar Bush, who's shown here, to develop a plan for the federal funding of science. Up until then, most science was supported by private foundations, but as a result of the contributions that science made to health, to armaments, to and to basically detection of foreign aircraft, the president wanted to make a bigger investment in science, and that investment was directed toward making basic discoveries using government money in our academic institutions, and the application of that knowledge to the development of practical goods in the private sector, a paradigm for funding science that persists to this day. The public support that was generated by his talk led to a transformation of the NIH campus, which is shown on the left in 1940 and now has been dramatically expanded over the ensuing years. And that was part of an ethos in which I grew up, one in which the federal government's support of science was considered to be a mandatory aspect of the way our country worked. Now, shortly after I entered cancer research in 1970, President Nixon, and I point out that science has always been a bipartisan activity in the U.S., supported by both Democrats and Republicans, and President Nixon, influenced by the strong advocacy for cancer research by many wealthy, influential people, signed an act that provided great resources for the National Cancer Institute and a determination in the U.S. to do something about cancer. In some ways, my own career as a cancer researcher has been bookended by, on the one hand, President Nixon signing this bill, and a more recent announcement by Barack Obama, for whom I worked for the last 5 years, announcing an initiative in precision medicine in the East Room of the White House. I'm sitting just a few feet from where he's speaking, and let me
the budget I sent this Congress on Monday will include a new precision medicine initiative that brings America closer to curing diseases like cancer and diabetes and gives all of us access potentially to the personalized information that we need to keep ourselves and our families healthier. You can match a blood transfusion to a blood type. That was an important discovery. What if matching a cancer cure to our genetic code was just as easy, just as standard? What if figuring out the right dose of medicine was as simple as taking our temperature? And that's the promise of precision medicine, delivering the right treatments at the right time, every time, to the right person. First, we're going to work with the National Cancer Institute. We want to find the genetic factors that can lead to cancer. And we want to use that knowledge to develop new and more effective approaches to help people beat this disease.
Now, there was a long intellectual distance, not just many years, between President Nixon's determination to do something about cancer, a disease that we didn't understand at all in a fundamental way, and the announcement by President Obama that we were going to make use of genetic knowledge about cancer, knowledge that was totally unavailable in 1971, to think about new ways to prevent and treat the disease. And in between, it was necessary for basic scientists to develop knowledge about a large number of questions: how the DNA in our cells encodes the information that tells cells how to behave, how cells know how to make proteins, the building blocks, the working elements of a cell. We needed to learn how to completely decipher all the genes, all the chromosomes in our cells, something we call the genome, how cells convert from a normal behavior to a cancerous behavior, and then how to build drugs and antibodies that could be used once we have this genetic knowledge that President Obama was discussing to make new kinds of therapies. So to tell you a little bit about how that happened, and both the culture and the basic knowledge required to develop an approach to cancer that's based on a deep understanding of how animal cells work, first necessary to begin to understand how it is that a cell, a very small element, but the basic element of our bodies, encodes information and uses that information to make the building blocks of cells. And starting with an understanding that it was DNA, desoxyribonucleic acid, that encodes proteins, and with an understanding that DNA is present in these entities called chromosomes that are observable in a light microscope, as shown here. Information then was developed to allow us to understand how the knowledge that a cell carries in its DNA is used to make the proteins of a cell and to allow cells of different types to behave in different ways. Probably everybody in this room knows that DNA is formed of two large, very long strands that are wrapped around each other in a way that we commonly refer to as a double helix. And that double helix is shown better on the right than in the more simplistic form in the center, has the four elements of DNA, A, T, C, and G, paired in a way that is extremely useful for copying DNA and reading its information. Knowing something about how DNA works and learning something about other large molecules in the cell, it was possible in the 1960s to begin to enunciate what is generally referred to as the central dogma of biology, that DNA has the knowledge, that knowledge is then put into a slightly different form, another so-called nucleic acid, chemically similar to DNA, but different in being only a single strand and some subtle differences in the molecules that make it up. And then that intermediate molecule, the RNA, is then translated, as we might say in literature, to make protein. It's useful to be metaphorical in explaining what this is about to an audience like this one. In a sense, a cell's DNA is like a large book. The book is the genome. It's the information that's encoded in those 23 pairs of chromosomes that contain DNA. Or when I do this kind of lecture with my son and his jazz band, we talk about DNA as being like a musical score in which there are only 4 notes. And when we think about DNA, looking only at one strand at a time, we can think of DNA's 4 basic chemicals as being like letters, A, C, T, and G, or notes. They are read as triplets, each triplet denoting one of the 20 amino acids, chemicals that are used to make proteins. And basically, the information you need to know at this point is here, that the information that's in DNA tells cells how to copy that DNA, because every time a cell divides, all the DNA must be copied. And there are a lot of elements here. There are 3 billion elements or letters, and they have to be copied accurately. And then that information has to be used to make all the proteins that are required for different kinds of cells. And there are lots of different kinds of cells in a human being. This slide just shows about a half dozen of them, red cells and nerve cells and blood cells of various kinds. But those of us who are looking carefully at the nature of cells in the human body estimate that there probably are at least several hundred and perhaps a few thousand different kinds of cells, and every cell has to be different, and it's different because of the roughly 20 or 1,000 so-called genes in a genome. Different genes are read out to make proteins in different types of cells. I want to show you a movie that illustrates the problem at hand. That is how each organism, every one of us, every organism that we know about in nature, has emerged from a single cell. So this is a miracle. If we think of a fertilized frog egg, for example, as shown in this movie, you can see this is one cell. It's an egg that's been fertilized by a frog sperm. It's dividing into 2 cells, then into 4, then to 8, and so forth. At this point, all the cells look quite similar. But with time, the cells begin to achieve a certain kind of distinction with respect to their appearance and their behavior. And that distinction is imposed on cells because different genes are being read out or expressed at different times. We're watching a speeded up version of development of a frog egg into a tadpole. Now you're seeing the spinal cord and the vertebral bodies of a frog being developed in a horizontal way across this growing embryo, and you can see it's beginning to form a shape with the head at your left end. The heart will soon become visible, and some eyes, and you can see the eyes appearing, and a primitive mouth. And all of this is happening by receiving the instructions that are present in a single genome. You see the heart beating in the center there. Now you see something looks more like what you imagine might be an early vertebral species. So that is one challenge met. We don't fully understand yet how that happens. We know some of the principles. Proteins that are in different colors, balls that represent different parts of the machinery that carries out a synthesis of the DNA, makes a beautiful illustration. This is work by the biologist and illustrator Drew Berry, who works in Melbourne, Australia, showing you the remarkable orchestration of these proteins that have evolved over millions of years of evolution to make a machine that unfurls the DNA, then copies it with a very, very high degree of fidelity. If it failed to do so, the new copy would be a mess, and organisms could not properly develop. So these are some of the things that science has taught us over the last 30 or 40 years, how this kind of synthesis of DNA occurs and the nature of the proteins, all of which are critical to the survival of species. Now, the next important question is how we make the intermediate molecule in the central dogma, RNA, from DNA, and that's done using another set of enzymes that are shown here. You can see attached to what you now recognize as DNA, a new protein machine that is jumping onto DNA at a place where a gene is found, a region which encodes and teaches the cell how to make proteins. The little yellow dots that are focused around here are the components of RNA, the molecule that the enzyme that is now coursing along the DNA double helix is synthesizing. And you can see these large streams of large RNA molecules being produced as this blue-colored machine courses along the DNA and basically spits out a very long chain of RNA. This molecule is being made in the center part of the cell, what we call the nucleus, and it will have to escape from the nucleus in order to allow proteins to be synthesized. And then we're going to see very quickly how protein is then made from the RNA. And you see an RNA molecule that is now emerging from the nucleus of a cell. It's coursing through what we call the cytoplasm of the cell, the the viscous surroundings of the nucleus, and it has another machine now attached to it, a machine that's called the ribosome, the machine that helps synthesize proteins. The proteins are made from small red elements in this picture that are called amino acids, each one of which is being carried by these green carriers that have a certain chemical distinction and encoding properties of their own. And as you'll soon be able to see, protruding from the ribosome after a period of time is this red chain, which constitutes a protein. In this case, the protein that carries oxygen in our bloodstream, hemoglobin. And this process, as you can see, is very rapid. All these pictures that I'm showing you show these processes of synthesizing DNA, synthesizing RNA, synthesizing protein at the speed at which these things are actually happening in your cells at this very moment. So every one of us has many billions of cells. They're all copying their DNA at the kind of rate that we've seen this process happening in these astounding animations. And it gives you some sense of how remarkable our own bodies are, and how, when understood at the molecular level, we get an aesthetic appreciation for how cells have evolved to work. And they're complicated, so things go wrong. Now, things go wrong even when our cells are dividing naturally, because imagine yourself copying 9 or 3 billion pairs of bases. It would, or 3 million letters from a text. You're going to make mistakes. And when these mistakes occur, they can, that may mean that you have replaced one letter in the book of life with another, or pieces of the book, chapters have become rearranged, resulting in rearranged chromosomes. And all these changes may occur during development and during adult life. And when those changes occur, they may affect the way cells behave. In fact, when those mutations affect certain kinds of genes that I've given my career to studying and identifying, genes that we know play a role in the development of cancers, the mutations of this kind can lead to cancers because they affect proteins that are important for the normal growth and mobility and other properties of cells. And it's that effort of identifying the genes that undergo mutations that lead to cancer, knowing how those genes work, what kinds of proteins they make, how those proteins behave, that has led to a new era in thinking about the diagnosis and treatment and even prevention of cancer. Now, for all that to happen, it was necessary to have much more powerful tools for reading the genome, for deciphering the genome, for determining the exact sequence in which all the letters of the genetic apparatus are present in DNA. A major point in our thinking about this problem came in 1985 when the great cancer biologist Renato Del Becco argued that we should begin to develop a way of determining the sequence of elements in DNA, both in cancer cells and in normal cells, so we can understand exactly what is changed in a cancer cell. And indeed, over the next 15 years or so, a large fraction of biological research was devoted to the idea of determining the complete sequence of the genomes present in a variety of organisms, including human beings. And by 2003, it was possible for these two revered journals in science to lay out essentially the full sequence of letters in human DNA, and to begin to give us some idea of how many genes there are, what they look like, how they're positioned on chromosomes, and providing a blueprint for human genetic information that has been pivotal in trying to understand a wide variety of diseases, neurological disease, metabolic disease, how the immune system works. But for the moment, we're going to speak to the problem that I've been concerned with and is a concern to many of you, that is the problem of cancer. And I'm going to tell you a little bit about how learning such information about the abnormalities in our genomes in cancer cells has changed the way we think about the disease. As I assume most people in this room know, traditionally, and I'm not talking about traditional Chinese and Indian medicines, I'm talking about traditional medicine of the sort practiced in most of our advanced hospitals. Cancers are treated by surgery, which is a great approach. If you can remove the entire cancer, you can cure it by using what's called chemotherapy, drugs that basically destroy all cells but preferentially target cancer cells, and by using radiotherapy to destroy cells by basically smashing up DNA. But I'd like to now draw your attention to the newer kinds of therapies that are made possible by our deeper knowledge of how cancer arises and how biological systems work. And I will both outline the major strategies and tell you a couple of stories about them. So there are two kinds of therapies that are now novel and unconventional. And the first is to use drugs, to find drugs and then use drugs that specifically target those genes, more accurately the proteins made by those genes, encoded by the genes, that are responsible for cancer. Let me give you the most successful, the most famous example, and then tell you about another one or two. Some years ago, indeed 55 years ago, investigators in Philadelphia showed that most patients who have a leukemia of middle age that happens to be called chronic myeloid leukemia have an abnormal chromosome that turned out to be made up of pieces of chromosome 22 joined inappropriately to a piece of chromosome 9. And that abnormal chromosome forms a fusion gene, a hybrid gene, that makes an enzyme that's dangerous to the cell. It drives the cell to grow too quickly and to fail to undergo cell death as it normally would. Over the course of just a couple of years, scientists at Harvard University, in conjunction with scientists at a drug company, found a molecule that would stick in it's shown in green, would stick to a place in the protein, this abnormal, dangerous protein, and inactivate the enzymatic activity that this protein carries out. And as a result, the loss of an activity which was driving the cell to behave like a cancer cell put the cell at risk of dying, and indeed these cells do die, and the patients go into what we call a remission. The cancer cells largely disappear, although a few remain behind, requiring the drug to be used for the rest of that individual's lifetime. Nevertheless, this produced a tremendous change, a disease that affects about 6,000 people in the United States every year, and about 15 times more people worldwide, can be controlled to allow these individuals to live a normal lifespan. Now, this drug and many others have now been used in a wide variety of patients in whom an appropriate mutational target of the kind President Obama was describing has been found. Let me give you one story. You're all story lovers about this. A story that involved a 10-year-old boy who had a leukemia affecting what we call B cells, a component of the immunological system. And this cancer was not responsive to the usual kind of therapy that has been developed for treatment of this disease over the last 30 years or so. This boy seemed subsequently to be about to die of his disease because we had no conventional therapy to treat it. But then molecular tests were done that showed that the patient had an abnormal chromosome, and that chromosome, very much like the situation I described for you with chronic myeloid leukemia, but different chromosomes involved, produced an abnormal protein which was inhibited by the same drug that works for the chronic myeloid leukemia. And once that was applied, it was possible to see a dramatic change in this 10-year-old boy's blood and in his bone marrow. He went into what we call a remission, no evidence of disease, and you can see this young man with his oncologist on the lower right, and his disease is under complete control now, about 3 years after therapy was begun. The other kind of novel approach to cancer therapy that's been made possible by the genetic revolution is to make better use of the immune system. The immune system is poised to recognize almost any kind of foreign proteins, viruses, bacteria, parasites, abnormal cells that carry proteins that have been changed by mutation. However, until recently, it wasn't possible to use the immune system effectively as an approach to cancer. But over the last couple of years, a couple of important things have happened. First, we have come to understand that there are both negative and positive forces acting on the immune system. And we now have antibodies that can block the negative forces and allow the immune system to respond much more vibrantly, much stronger to the minor abnormalities that are present in cancer cells. The other thing we've learned to do is to actually change the words that are in a genome, to edit, to use your own language as writers and editors, to edit the genetic information that's present in the language of a cell. And although this is all quite new, just in the last couple of years, reports are beginning to emerge, such as the story about this young child, 10 months old, who also had a so-called refractory leukemia that was not responding to conventional therapy. And it was possible to take cells from another individual and to use gene editing methods to rewrite what's in the genome of the cells, the immune cells taken from another individual, so that those cells would then respond to the leukemic cells in this individual's blood without attacking the individual's normal cells. Now, this has not yet been through the traditional peer review. This picture comes from Science magazine last week, showing this child who now has a shot at being cured thanks to these genetic editing processes and the understanding of the immune system, has a shot at actually being cured. Now, I'd like to spend the last few minutes of this talk saying something about another kind of information. All of us who are scientists and are enthusiastic about the work that we do want the work to be known by the larger public, folks like you, by our fellow scientists. We don't write for money. We write for fame. We write for appreciation of the work. We write for the ability to disseminate information so others can build on it. Now, one way to do this is to have people who understand science act as science journalists and report on major changes in our own fields. This slide shows two important books published in the last several years. One of these books is written by someone whose family came from Calcutta. He's Bengali, a good friend of mine in New York named Siddhartha Mukherjee. And his book, The Emperor of All Maladies, highly recommended, wonderfully written, gives a rich history of cancer research and the new potential. It's been made into a movie for public television that if you have 6 hours to spare, you would enjoy watching. The other is a book called The Cancer Chronicles by a friend of mine who writes for the New York Times named George Johnson, who tells the story of his wife's battle with ovarian cancer, which he helped her conquer. Subsequently, she said she wanted a divorce, but they remain in good terms. And much of this has to do with a general attitude that I would like to think my scientific colleagues share. And that attitude is expressed vividly in this painting by Rembrandt, showing a Dr. Tulp from Holland, who in the early days of the 17th century would bring his fellow citizens in to learn about the nature of human anatomy. Now we'd be more interested in telling you about human genes, but he was gracious about bringing his fellow city officials into an autopsy room and showing the elements of the human body and how they work together and why they might fail. But it's this attitude to share learning that I believe ought to underlie the way in which we as scientists disseminate information to others. To do so, we require some changing in the culture of science. There are two great traditions in the curation of information. One on the left is to distribute books to the public, as exemplified here by the great public library in my own city, the New York Public Library. And the picture on the right shows a tradition I'm much less enamored of, the monkish tradition of having great books locked up in monks' chambers where the acolytes of the discipline close the doors to the public and work hard on these sacred texts. There are at least three ways in which my colleagues and I have been trying to change the culture of restricted access that has been imposed by a publishing industry which has made many valuable contributions to this dissemination of information, but in order to recover its costs has been imposing considerable subscription fees on those who would like access to the information. So there are three things that we have been doing over the last 15 years or so that are changing the public's access and our fellow scientists' access to new knowledge about science. The first is to build so-called public libraries of science that depend on new digital methods. So, for example, at the National Institutes of Health, we've created something called PubMed Central, and anyone anywhere in the world can see now about 80% of the literature published in the United States within 6 months to a year after publication, simply by putting the authors' names into a dialog box and looking at these publications, which are there in full glory. The second development was to develop a new way to publish science so that the expenses are covered not by people who buy subscriptions, but by the authors who use their grant money, a very small fraction of the amount of money that's required to do the research, to pay for the publishing costs and to make the papers immediately available at the time of publication, fully accessible to anybody. There are no subscriptions. You simply go online to the journal and the papers are there. And there are now many journals of this type, the Public Library of Science journals that I was involved in starting about 12 years ago are among them, and now about 20 to 30% of our literature is immediately available. And the third that we're now exploring and with a lot of energy is the idea of providing yet quicker access to the discoveries that have been made by putting our articles online before they're officially published and reviewed so that people can see the work that's ongoing even before it's been officially published. And the reviewing can be done online with open access to the reviews, and papers can be incorporated into the public libraries once favorable reviews and appropriate changes have been made. And this is a way to accelerate information and get it into the hands of people who can build on it much more quickly. So I've tried to tell you about how politics and science are interacting. Tried to tell you something about the incredible ways in which we've now come to understand how cells work. I've tried to tell you something about how our knowledge of biology is influencing our approach to treatment of cancer. And I've tried to tell you something about how we, in common with you, attempt to publish our work and make it available to those who are interested. For those of us who do cancer research on a daily basis, as I still do, there's a lot of work ahead. We know that the ideas that I've told you about new kinds of therapies based on developing drugs that are targeted to certain proteins or based on new understanding of immunology, still far from complete. And the precision medicine initiative that President Obama has endorsed still has a long way to go. We need to pay more attention to cancer prevention. I and my colleagues believe that we could probably reduce the number of cancers that are occurring throughout the world by 50% or so without too much difficulty by controlling smoking, using the new vaccines that have been made against viruses like the human papillomaviruses more widely, by reducing exposure to ultraviolet light and to certain kinds of carcinogenic information. The third big approach, one I would love to talk about in more detail here than I have time to do, takes into consideration the recognition that causes of death in the developing world are moving away from the traditional infectious agents like the diseases of malaria and tuberculosis and AIDS to the chronic diseases that are also common in more advanced economies: cancers, cardiovascular disease, chronic lung disease, diabetes, and metabolic disease. And many of us believe that cancer is among those diseases that can be much more effectively controlled through prevention, through treatment, especially inexpensive treatments, and through the improvement of health systems in a wide range of countries, even the very poorest countries in Africa, but certainly in many of the advancing economies of Asia, South America, and that global approach is a very critical one that has a major role to play in the work that we have ahead of us as cancer researchers and advocates for the use of what we know about cancer to improve public health. Thank you very much for listening to this story, and I'd be more than happy to take questions now or on Saturday when I'm going to be taking an interview with Sadif Saez. Thank you very much. Now, can we put the lights up? There are microphones, and I see some hands already raised, and I will take this out. Can we provide some of these, those with questions with a microphone so everybody can hear the question?
That was a great talk, and you know, you've covered 3 different themes there with politics and then communication and the research itself.
You were warned in the program this would be a diverse talk, many topics.
Yeah, but my question is, you know, when you talk about politics and government, immediately it comes to my mind bureaucracy. I mean, as you know, the U.S. is far ahead in doing research, not only in just cancer cure, but all the other diseases out there. And in the recent case of the Ebola thing illustrates the point I'm trying to make, with the FDA trying to have this elaborate process of approving drugs, sometimes can impede the use of those drugs by people who need it very urgently. So can you speak to that?
Yes.
How can we manage that better?
Fine. Okay, well, first of all, I'm going to challenge your idea that the FDA is at all responsible for the fact that we had so many cases of Ebola, but I will tell you something about that. Let me say, first of all, just a word of background. I did talk, and we can have the lights up. I'm not going to show any more movies or slides. I know it's very, very murky in here, rather smoky, and that's not my fault. I've not been smoking anything up here. Good. First of all, I've made a point that presidential endorsement of science is very useful in America, and the growth of the National Institutes of Health very much dependent on presidential support. But over the last 12 years, the NIH has been losing spending power, the change in the economy, some loss of interest in science among those who lead our government, a very high level of divisiveness between Republicans and Democrats has impeded the growth of scientific support in the states. There are many students who now would rather go into financial industries than into science because they see their mentors struggling to get grant support. All is not well. And we do have a bureaucracy. It's a bureaucracy that has made universities required to provide a lot of additional administrative services to be sure that the bureaucratic oversight of science is carried out in a way that doesn't lead to penalties for the universities. So we have some serious problems. Now, you're alluding to some inhibition in the speed with which certain kinds of therapies or preventive strategies are developed. In the case of Ebola, there's an important story to be told, and that is that colleagues of mine over 10 years ago had developed the first steps toward an Ebola vaccine. And this is a good story because they understood the principles of vaccination. They knew that Ebola was a very deadly disease, and they had information that would suggest that the vaccine was going to work. But that's as far as it got in the academic sector. At that point, at least in the US system, it's required for commercial entities, pharmaceutical houses, even biotech companies, to pick up a project. And at that point, there had been no examples of Ebola outbreaks that had spread beyond small villages. Only a few hundred people total over the course of many years had died of Ebola. And no commercial entity saw a market for an Ebola vaccine. So it was not developed by industry. If we had had an Ebola vaccine, we could have controlled that outbreak very quickly. But there's something in our system that says everything has a cost, and if you're a commercial company, you want to profit for that. Ebola is one of several very deadly viruses that has caused occasional outbreaks. There's also the Marburg virus and several others. And companies that have to make a commercial decision about what to work on just simply did not pick up that vaccine. Now it is being further developed, and I believe the next time we have an Ebola outbreak, that will be available. The drug you're referring to is still of questionable efficacy. The vaccine is going to be much more effective. What proved to be effective this time, of course, was public health measures that were difficult to institute in the 3 African countries. As you know, there was an hysterical response. One man came back to New York City, my town, and people were refusing to ride on the subway because he had gotten on the subway to go have an evening at a bowling alley. People behave in an irrational fashion at that point. So there are many public health messages, but the message I would not take home from this is that the FDA is a negative force in this system. Indeed, the FDA has developed swift methods for approving new drugs and vaccines. It is a bastion against the development of unsafe drugs. It's helping developing countries produce a drug industry that eventually will be selling more drugs in the United States, hopefully at lower cost than we currently pay. So I'm a friend of the FDA's, and I don't think that is the thing we need to worry about in the American bureaucracy. There are many bureaucratic flaws that I would criticize, but that's not one of them. Yes?
The problem with science, one aspect of it is that there is this element of certainty. Now, with the medicine, you go through a process, you prove it, and then you set it out in the market. With respect to genetic modified drugs or crops, for instance, you have the relationship in the US has one perception of genetically modified crops. They've let it go outside. It's not the same in Europe. They're far more reticent about it. And that's a question of, it's not just politics.
Of course it's not just politics.
There's something more to it.
Yes.
That's one part of it. The other one is the aspect of the finance. The government can provide you all the support for research, etc. The relationship is when you depend on the market. Now, your example about Ebola, that the NIH had developed the Ebola virus vaccines early on, and there was not enough cases. It's not the same with respect to malaria. America had done an enormous amount of research during the Vietnam War. As the war toned down, American companies particularly, because the research in medicine, etc., is done far more by the private sector than the government itself. I stand to be corrected on that, but as the Vietnam War ended, so did the American companies decided that there's no market, although the incidence of malaria is widespread. And there was little research done in malaria, and yet we're still struggling to find a cure for malaria. Two
Well,
aspects.
all right, you're raising, this is more of a comment than a question. Let me just say a couple of things. About malaria research. Now, malaria research, it's true, went into a recession largely because of the use of insecticides around the world. And the incidence of malaria fell dramatically in the 50s and 60s until it was determined that the use of DDT and other sprays to kill the Anopheles mosquito was a mistake. And then malaria has resurged in Africa and India and many other places. We do have good treatments for malaria, especially now that the artemisinin are in wide use. Malaria drug resistance is a problem. There are some answers to that. We don't yet, I agree, have a good vaccine. We have one vaccine that is of intermediate value. I don't think it's so much retreat because of the war, but I myself have been heavily involved in the 1990s in trying to bring malaria research back into a well-supported condition, which it now is. I would argue that malaria actually is being quite well supported, and through better use of bed nets, better drug combinations, possibly even the development of this partially effective vaccine, that malaria is under much better control than it was 15 years ago. I agree with you. I've made a very limited foray into the politics of science. There are many other aspects of the politics of science, cultural wars about how we make use of advances. I personally believe that we should be, while we're cognizant of the fact that making genetically modified foods needs to be studied, so far I don't know of any evidence that those modified foods have caused any medical problems. We don't want to be denying people in Africa access to fortified foods and more abundant foods and better crops for their soil simply because someone in Europe doesn't believe that there should be a piece of foreign DNA in a food that they eat. So that's a complicated discussion, which we probably don't have time to get into in more detail, but I think that there's been an overreaction against genetically modified foods, when in fact we've been genetically modifying foods for thousands of years by making much less well-controlled crosses between subspecies of plants and animals, and not really in a much less controlled way than we do when we use genetic engineering.
Using a social determinants approach to health, the APHA, the American Public Health Association, talks about health in all policies. And how feasible do you think that is?
Well, I think it certainly plays a role in the approach that the U.S. is making and that the UN is making to development goals. I think over the course of my own lifetime, we've gone from a time when people was saying somewhat cynically that if you extend life without improving economies, that is actually bad for a country. I think we all now realize that healthy people make for a more productive economy, and that in fact better health means a rise in standard of living. As long ago as 2000, the UN sponsored through the WHO a volume that I contributed to on the macroeconomics of health, which laid out a formula for things that could be done to reverse the most reversible of the maladies that affect the poorer parts of the world, and indeed the development goals of the UN that were initiated in the year 2000 featured at least 3 important health goals. More recent developments of the UN include efforts to bring the power of modern science to combat the chronic diseases that I was mentioning at the end: diabetes, lung disease, cardiovascular disease, stroke, and cancers. And I do think that I feel very strongly that institutions like the NIH and other large funding agencies should be spending quite a bit of their resources to developing better ways to prevent and treat disease in poor countries. Yeah.
Professor Varmus, how can we use policy to make science a more attractive career choice for young people? Because this is a huge concern for countries all over the world.
Right. No, I agree with that entirely. I think you have to tailor your approach to the particular country in which you have your concerns. In some countries, science is not part of the culture, that is basic science, and you need people to come and explain to policymakers, to finance ministers, and to others that science has cultural value and it has economic value. In a country like Bangladesh, for example, one doesn't need a whole lot of schooling to appreciate the effect of climate change and the rise of the oceans on the living conditions in this country, and the need to make a contribution to research on energy and climate change and environmental changes is going to be of benefit to this entire region. In the U.S., we have a problem with recruitment of the brightest young people into science, and it's to a very large extent a result of the perception and actually the fact that it's become increasingly difficult to try to maintain laboratory activities. Science is expensive. NIH funding, for example, has been falling. Science funding in general has been declining over the last 15 years or so, and young people see a much greater chance of financial success in financial services, in the computer industry, and I think it's incumbent on those of us who are senior scientists to try to develop special training and grants programs that improve the way in which we conduct our science. A small group of us have recently written articles about the task of rescuing biomedical research in the U.S. By trying to find better training programs that allow people to become independent investigators at an earlier age, by developing new ways of doing business, keeping lab sizes to a relatively small size, and make better use of shared resources to make the whole enterprise more efficient, more economical. These are not easy things to do, but there is tremendous interest at this stage with the scientific enterprise in the U.S. being a much more difficult environment in which to prosper, and I think that we have the engagement of, especially of young faculty and trainees, to try to make our scientific enterprise work better. In other countries, the problems are sometimes different. Certainly, Europe practices science somewhat more differently than we do, but and I don't know enough about what's happening in South Asia to know. I simply know that from being in India a fair amount in the last few years that India is making increasing contributions to the financing of science. They're finding useful ways to bring people who've been trained in Europe and the US back to India. They're creating new research institutions. They're making science seem like an attractive enterprise through TV shows and other means. Certainly in the US, we're making an effort to popularize science in certain ways, science fairs. Perhaps that's the next challenge for Dhaka, is to have a Dhaka Science Fair. In New York, we have one. Actually, in New York, it's called the World Science Fair, that brings hundreds of thousands of people on the streets and into meeting halls over the course of a week to hear about developments in science and hear about the excitements and beauty of science. Television is a medium that can be exploited, especially now that there's so many outlets through, especially some of the cable channels, for describing science in ways that's attractive to students. So there's no simple answer to your question, but having lived a life of science after starting out as someone interested in literature and have no regrets, I'm always happy to put my oar in to attract students to what I think is a remarkable career with a lot of practical consequences for the way in which our society functions and prospers.
Hi. Hi.
Can we put the lights back up? I don't know why they keep dimming. In fact, the air seems to be clearing remarkably. I don't know. Okay.
Hi, my name is Promit, and
Where are you?
I'm here.
You're here. Where's here? Oh, yeah. Okay.
Okay. Yeah, my name is Promit, and I'm an oral surgeon, and I'm currently applying to grad schools because I want to be an oncology researcher, and I'm primarily interested at the disease multiple myeloma. So my first question would be that there are still diseases like multiple myeloma. There are several types of leukemias, and there's pancreatic cancers, which is still incurable. We cannot still treat refractory multiple myeloma. We still cannot treat several versions of leukemias, and we still cannot treat the pancreatic cancer. So would you mind putting on some light as to why they're still incurable? Sure. And my second question would be looking back, you have been rejected twice by Harvard Medical School. So when you look back in retrospect, do you think that it was a boon or a curse to your career? And there's something I would like to add, that since I want to be an oncology researcher and I'm currently applying to grad school, can I talk with you about a bit about my grad school plans outside of the stage?
Well, I'm going to ignore the last part. You can try to see me privately tomorrow or the next day. I'll be here throughout the festival. Did it matter whether I went to Harvard Medical School? No. There are many good medical schools to attend, and it actually is a good story to dine out on, being rejected twice by Harvard Medical School. Thank you for reminding me. Let me say one serious thing, though. I hope I didn't mislead the audience. I told you some exciting stories about successes with immunotherapy and targeted therapy, but in general, most cancers that progress to a very advanced state ultimately kill the individual. We've had a lot of success in extending the life of patients with metastatic cancer. We have learned to control symptoms. I didn't talk about that, but controlling pain through the appropriate use of opiates, controlling infections, controlling the suppression of the blood system. All these have made living with cancer a much more palatable episode in someone's life. But as you're pointing out, there are many cancers that prove to be still resistant to the kinds of therapies, even the new ones that we've developed, and that is by far the more common outcome of cancers that are advanced. And in the case of myeloma, the disease that you're interested in studying, we have certainly prolonged the average life expectancy of someone who has been diagnosed with myeloma from a few years to 7 to 10 years through transplants and some new drugs. But it is a lethal disease. Why is that? In part because of the nature of the cancerous process. Cancers continue to evolve. They develop new mutations that render the cancers resistant to drugs, resistant to immunotherapy sometimes, and we haven't figured out how to control that. The answer is probably going to rely on stronger drugs, drug combinations of radiotherapy and drugs or immunotherapy, but we don't know how to put those instruments together in the most effective way. We also have many cancers, such as pancreatic cancer, one that you've mentioned, that in virtually every case is driven by essentially the same kind of mutations affecting a class of genes for which we don't have any targeted therapies at all. How do we deal with that? Well, one thing that I did at the National Cancer Institute my last 5 years there was to build a nationwide effort to try to combat mutations in the genes that cause pancreatic cancer, and same genes are involved in about half of colon cancers and about a third of lung cancers. And it simply requires more science. We have some new clues to how we might combat those, but I don't want to minimize the very large problem that we have remaining. We have a much deeper understanding of cancer. We have new tools. We have tools that can be sharpened, but there are new things that remain to be discovered if we're going to control these diseases more effectively. I don't know who's in charge of the session. I know there's another one after it. I don't want people to get too far behind. But if you tell me how many more questions to take, let me take the lady in the orange in the back. I can't hear you. Turn the microphone on, please.
She says it's on. There we go. This has been extremely interesting and informative indeed. My question is, could you comment on the role of big pharma in the politics of science?
Sure. I mean, I'm not sure it's the kind of politics I was addressing, but let me just say a word about big pharma. The large pharmaceutical industries, the large companies, have done some extraordinary things in developing new drugs and vaccines that are very effective in treatment. The course of developing a new drug is a long one. As someone mentioned a moment ago, in the aggregate, industry spends more on medical research than the government does, or the government plus philanthropic donors to academia. And yet much of the research, of course, is very pragmatic: clinical trials, medicinal chemistry to make drugs better. The vast majority of basic research is still done in academia. The main complaint that people have about the pharmaceutical industry at the moment, although I think there are a number of things one could complain about, but the major complaint is the high price of drugs, a price which is extremely high in the US especially, less high in countries that have price controls, which we do not have. And the industry will say, with some justification, that drug development is very expensive, and to recover their costs, they need, especially to recover their costs in America, where prices are not controlled, they need to charge prices that are essentially unaffordable for people without appropriate levels of insurance. So that I find difficult to deal with when people have to, under the insurance plans that most people have to use their life savings to get access to drugs. So in part, the problem is not solely the drug companies. At least in America, the problem is in part that we don't yet have the kind of health insurance that I would like to see. I think health insurance should never exhaust someone's personal resources, that we ought to have better coverage, and whether we go to a single payer system or not a topic I want to debate here. There ought to be a better way to cover the charges. Controlling drug prices is a whole other problem. The high level of cost, of course, is temporary, because eventually patents run out and generic drugs are made at a much lower price. But we do have a new element in the drug industry, because many of the new so-called drugs are actually biologicals. They're antibodies or cells or entities that are very difficult to reproduce, and it's going to be harder for generic forms of those drugs to emerge and provide competition in the market. So I don't have a simple answer for you, but if we want to have a drug industry that's working hard to make these difficult new drugs, we're going to have some financial incentive. How we get control of that is going to depend a lot on how each country develops plans for covering health costs. Okay, thank you all very much. I don't know why the lights can't stay up, but thank you a lot for the questions.